in Omicslogic community, as I said, it's a free community. Anyone can join for free. Here you can see register for free. But there are premium courses which requires you to utilize the T-Bioinfo server. So there's a subscription for the program, a very minimal cost that we have kept together. And that's why we are calling it a special opportunity for you to learn from us and learn from the resources that are available. So you can join the program to access the premium courses. Otherwise, you will have access to the free courses. I will tell you which free courses and how you can access those as well once you have created a login. I'll do that. But before that, I mean, if you are interested in going through the entire, sessions, going through the workshops by experts, going through the premium courses and all of that, then you have to join the program through this page. And it's kind of simple. So let me show you. I'm already a part of the program. So if I have to join the program, how can I join? Yeah, obviously you'll have to log in from your account first, and then you will see the program page. And once you go in this program page, or if you can also like go to the link directly, you will be able to come to this program page, and then you will have to complete your registration. So you can give your information, and we will be sending you more information about the program, more details if you are not sure about what will be covered. And at the same time, you can start your application and you book your seat. So it's a program that will be available for anyone to take. And you can join this entire, whatever we covered in today's session will be available with the beginner's license. So we have different subscription levels. So the first beginner subscription level is just for $10 a month, which will give you access to all of the things that we discussed today. And we have different other subscription levels for you to do research, for you to do work on project, and maybe we will discuss it in the next time when we meet. But for this program, you will be able to do all the coursework and all the live sessions that we just discussed. And about the coursework, I'm going to tell you what all coursework are there, which are for free and which are for premium, and why they're premium and why there's a cost. So let me go back to the slide deck. Yeah, the first course that we have put together for everyone to take is a free course, other than the ones that you see in Star are the premium ones. So when you do this course and go through this course, Bytes and Molecule, you get an overview of a very basic understanding about the different data types that we will be discussing in the program. So anyone coming from a background without knowledge of biology can start taking this course, Bytes and Molecules. So let me just quickly show you a video for the same.
Bytes and molecules. Every living cell has its own hard drive where it stores bytes of information needed to function and grow, adapt to environmental changes, and fight off dangerous intruders. This information is stored in molecules that are arranged in sequences of letters, referred to as DNA code. In the 1990s, a tremendous effort by governments, researchers, and companies resulted in the full sequence of the human genome. Since then, we've seen an exponential growth of molecular data that describes cells, tissue, organs, and organisms. Massive data sets containing genetic information about the young, old, healthy, and sick are now freely available to the public. Here, we will explore the various omics data types as well as examples of the information they contain, genomic variants associated with ancestry, gene expression levels responsible for cancer spread, and protein modifications that can render important code unreadable. This course is intended for all those interested in understanding how the current revolution in biomedical data impacts biomedical research and human health.
So as you learn from this is the first course that you will go through. And it's a free course anyone can take. And in this course, you will learn about the different data types. Another free course that we have is the Introduction to Bioinformatics. It's a course that takes you through different types of application of bioinformatics, big data bioinformatics, as I said. And it's a course where we get to learn about your expectations, what do you want to learn in bioinformatics, which area of research that you are interested in. So this course has a form. When you start this taking this course, you will get a form, and I'll request to Sonalika to share maybe the link to this course as well. So this is an introductory course where you will get the overview of different type of uh, mm, uh, fields where you can apply bioinformatics, and you can also tell us about your area of research where you are interested in. So these two courses is something that we would want you to start with, even if you are joining the program, even if you're not joining the program, it will be really helpful for you to go through these courses. So once you go through these courses, we also have other free courses, introductory courses are for omics bioinformatics. As I said, that in this program you will also have opportunity to learn coding. You will have also have an opportunity to learn this from the browser itself and get points and recognition and all of that by while completing this coursework. So in the this first tutorial, which is just to get started with bioinformatics, it tells you about loading structural data into our multidimensional data visualization in our reading very basic file, DNA file, and then understanding the DNA replication and reverse complements in R. So this is here also you will have free courses where you can learn about these different technologies and how it can be applied, different programming languages, how it can be applied for data visualization. Now, I mean, I told you about the premium courses as well. So the premium courses, how they can help you do the hands-on analysis on yourself by yourself at your own pace. So let's take a look at that. Let's have a demonstration about that as well. So let's go to the Learn portal again. And now what we will do is that we will go to the section called courses, and we will find all the courses that we are interested in. If you are interested in Python, we can try Python. If you are interested in R, we can do R. If you're want to learn about the introduction to bioinformatics and genomics and then epigenomics, then all of this course material is here. We will just have to search for them, and I'll show you that's pretty simple. Okay, I'll have to log in first. So I'm logged in now. I can see the courses. So let's take a look at the R course. So we have different courses, as I said, getting started with R. Another course is Introduction to Data Science using R. Now we have a lot of lessons within these courses that you can take and earn points from. So let me show you how it works and how it is so simple and easy to do this, that anyone can learn from this. Because when you start from basic, you tend to get an understanding of what's going on, and you understand different type of commands that are being used and get familiarized with it. So we start with you can install R locally on your computer as well and try this out, and you can do this from the browser itself as well. So with every tutorial that you see here, the first part is about is to understand the problem, what the problem is all about, what we are doing here. So we are looking at different type of data, categorical data or continuous data in data science, and then we are trying to do some plotting to see the characteristics of data. I mean, how they are placed, if we can separate them or not. So this is being used in many of the bioinformatics application, especially when we are doing big data analysis. So here, I mean, you will learn to upload the data, how you can use links and upload that data into the browser, and then you can run different sort of commands to analyze that data. So once we learn about these different commands, which is just shown here, you can go through them and easily learn about them. Then there is a breakdown of code that will be used for this tutorial. So what this code is doing, so it actually explains all of it and what kind of results that we can expect, and what kind of analysis that we can do from this sort of data. So once you do this, you get to a point that where you can practice it yourself. So it's a code playground where you can write your own code, and you can load data and you can run the analysis here itself to show that if you're getting the result or not. As you see, as you can see that this is already a code that is put together and is working. You can change this, and you can run your own code and do your own analysis, downstream analysis, using this code blocks, and different tutorials that have been shown here, uh, in the learn portal that you will have access to. So the opportunity is, there is a lot of opportunity to learn about different things. So I just told you about this R code. Now let me go back to the slides, and I'll show you what all different courses that we have put together for you to take. So you can start from Python as well, similar courses. You can apply and see what is the difference, how you can learn both of them, and choose what you are more comfortable in, and what kind of analysis that you are into, and what kind of research that requires to use these languages and apply these languages for data analysis. So another important aspect of this sort of learning, Omicslogic bioinformatics training, is to be able to access the T-Bioinfo platform and run your own analysis and get the structured data to analyze further in R and Python. So when we go through these courses, such as Genomics, as I said in the second session, after the introduction session, we will be doing Genomics. So in Genomics, we will be going through very basic, starts from the DNA structure and variants, why we are using the genomics technology, and where it has been helpful, how we can identify variants and identify mutations that are critical. So, learning about that, working using R, then introduction to genomics, where you get an overview of all of that in much more detail and depth, and then practicing using the pipelines. As I was telling you, that you will be getting an overview and understanding of how algorithms are connected together, and they give us a structured result after doing a complete analysis. So there are several tutorials, and the ones that are starred uh, includes practical assignments. So for that, you will be able to access the T-Bioinfo server to run some example pipelines. And now let me take you to the T-Bioinfo server and show you how it works and why it is so easy for anyone to use. I mean, even students without a background in bioinformatics and computational, computational sciences can do this. So this is the T-Bioinfo server, and once you register, we will be sharing the access for you to take some of the example pipelines from here. And now let me show you an example pipeline from the transcriptomics data analysis. So it's an AI guided platform, and we have put together examples from publications that you can take and learn from. And at the same time, you learn about the logic. So many of these algorithms that you see here are open source algorithms. So our purpose is not to make you dependent on a platform for data analysis, but make it simple so that you understand how the algorithms are used, what are the outcomes, what are the outputs that you get from this sort of analysis, and how to interpret that result. So understand the logic behind the analysis is what we are focused on. That's why we are called Omicslogic. And to do this, I mean, we have put together many examples for you to go through. One of the examples that I'm taking you through is the cell line data set. So it's an RNA-seq experiment where the scientists or the researchers have generated data on cell line, using cell line, breast cancer cell line samples. And then we are processing the raw data here using these pipelines. So we have already put together different sections for different sort of analysis. For example, pre-processing, mapping, quantification. These are different sections that you have to go through to process data and get a structured table. And now one thing that you can see is that they are differently colored. So to make it more easy and simple, they are color coded so that you understand that these sections are used for pre-processing, these sections are used for mapping, this is for quantification of expression level. And what are all of these steps? And we will be going through in detail in the workshop session. And also they are described, I mean, absolutely amazingly in the courses that you will have access and you'll have the opportunity to go through in this training. So the input file and the output file are already stored because it's an example pipeline for you to learn. So the first step that we do in such analysis is to clean the data, make it more structured. So we use algorithms such as Genomic, PCR clean, and then I'm going to show you how we can map it to the transcriptome and then generate a table of expression. So if you can see, I should be a little slow, I know, but it's a quick demonstration and we'll be going through this in much more detail in the session, and it's described in the courses as well. But here also you will be able to learn about these pipelines, these algorithms, what they do, what paper that you can read about them also, and learn about them in much more detail. So here, what we did is that we pre-processed the data, we mapped it to a transcriptome, and when we say if we are mapping or aligning it to a transcriptome, we are doing it on a reference transcriptome, and then we are generating a table of expression after doing the alignment or the comparison, and then we are ending the pipeline. And here you will see that there are different courses that you can take and go through, which are on the Learn portal. Now during the session we will be able to tell you the output and how to analyze these results and what is the outcome and what can you do next. So the next step is to take this data structure table, run some other analysis, and do some machine learning analysis based on the research that we are going through. So this is the way we kind of go and explore the Bioinfo platform. And obviously it has also the capability to do the analysis right from NCBI. So you can upload data from NCBI, you can upload data from your own research lab, and those are available with different subscription options to access this platform. With the program, you will be able to use it for educational purpose. You'll be able to do example pipelines and learn first before you do your own experiments. So that's the process that we follow for you to go through the technology. First go through the steps, do the data analysis on example projects, and then learn on your own or public data that is available, and that we discussed that it's so huge that we have been able to do a lot of quality research out of that data. So this is about the Bioinfo platform that you will have access to, and this is the coursework for genomics, and then we have the coursework for metagenomics. It's very elaborated coursework. As I was telling you, there are examples here that you can learn from, and learn about the OTUs, learn about different types of metagenomic analysis, learn about the data to pipeline, phyloseq. All of this is covered in different tutorials under this section. So if you go to the course for metagenomics, you will find all of them bundled together that you can take one by one and learn about and apply this to your own research once you've been able to go through this material, which is mostly hands-on and also gives an overview of all the technology. And there are also videos in the tutorials, so you will be able to go through the videos, recorded videos, and replicate that using the Bioinfo platform, because at the same time you will have an access to do this hands-on thing. So that's why this Omicslogic training is a really unique opportunity to learn for real. So the next technology that we will be covering is the transcriptomics technology, and we have detailed explanation of this transcriptomics data analysis. We've been doing several programs and have a lot of participants who have gone through this training and come up with unique projects that have been published, as well as got recognition in various conferences and presentations. So another aspect of doing an advanced level analysis is to learn about the application of machine learning. And we use different type of machine learning regression. So there are factor regression analysis, supervised machine learning, unsupervised machine learning. to kind of get insights and results from transcriptomics data analysis. Another advanced section that is again available, I mean, as I said, it's a subscription based model. If you are into this, then you can go in deep into this and maybe, try different courses, run some ideas that share with us some research ideas that we can work on. So there are different ways we can collaborate and do research together. So single cell transcriptomics is such a area, very promising area. A lot of modern research and high value research is coming out of, high impact research is coming out of single cell analysis. And there's a lot of data that has been deposited as well. So another aspect of doing this, all of this, is to learn machine learning, how we can apply machine learning, bioml. That is also is in there in the coursework. So you will also have opportunity. I know that there is a lot of coursework that you can do and learn from. So you can also take your time and go through this analysis at your own pace, and at the same time learn from the video that will be recorded and provided to you. And we will have also mentors that you can connect with and learn about specific sort of analysis. All those opportunities are available on with us for this Omicslogic training. So that is about the coursework that is there. Other than that, I have already told you about the data science part for R and Python, and then we have mentors and then we have programs, and which I can show you actually quickly here on the website. So when you're logged in, you have option to do coursework, you have option to select a program, and you have option to do research. So we believe that it's best to start with the coursework, go through the examples, and then conduct your own research and learn from specialization programs. One of such program is this one, that is Omicslogic, Bioinformatics and Computational Biology. People interested in infectious diseases, data science, transcriptomics, we have specialization programs that we keep on doing as well for those. But to those who have just started, this is a best opportunity for you to take Omicslogic, Bioinformatics and Computational Biology. So that was about